Let's now take a closer look at a prokaryotic transcription unit. That's what's being depicted here. A prokaryotic transcription unit, you remember, is going to have a promoter element and then a coding region. Okay, so remember that the promoter region is where the polymerase is going to come in. It binds to this promoter region. These are like the landing lights in an airport. The polymerase is going to come in. It's going to look for where to land. It's going to see these promoter elements, and boom, it's going to start transcribing at the +1 sequence. Now on this slide, I'm telling you that transcription's happening from left to right because that's the way the arrow is going. Now you should be able to tell me which of these strands is the template strand. The template strand is going to be the one that reads from 3 to 5 prime the same direction as transcription is happening. So the bottom strand here reads 3 to 5 prime, and transcription is happening from left to right, so this is the template strand. That automatically makes the top strand the coding strand. This is the strand where you see the TATA sequence and other promoter elements. Notice something. There's these areas of the DNA which are called 5' and 3' UTRs. And what UTR stands for is untranslated region. What you should recognize is that the UTRs, or untranslated regions, contain absolutely no information that will go into amino acid sequence. These do contain areas which will be included in the messenger RNA, but that information will never be seen in amino acid sequence in the protein. So the RNA polymerase comes along, it starts transcribing at the +1 site, it's going to read the template from 3 to 5 prime until it reaches a termination sequence, at which time the messenger RNA will be released. That is how transcription happens. So at the end of transcription, you're going to end up with a messenger RNA that goes from 5' to 3' end, goes from 5' to 3' end. At the 5' end, there's an untranslated region. At the 3' end, there's a 3' untranslated region. The area in between is referred to as the coding region. The coding region. That is the region of DNA that's now been conveyed into mRNA region. This is the important region that contains protein or amino acid information. It's the coding region. So, some facts about prokaryotic messenger RNA. Number 1, it may or may not be polycistronic. Let's define what that means. mRNA that's polycistronic contains more than one gene's sequence in that one particular messenger RNA. So there may be up to 3, 4, 5 separate genes encoded by one single messenger RNA. That's what polycistronic means. The word cistron is just an old word for gene. So polycistronic means many genes on one messenger RNA. No introns. That may not have a lot of meaning to us yet because we haven't talked about introns, but later on we will. Prokaryotic messenger RNA does not contain any interrupting or intervening sequences within the coding region. And finally, transcription and translation are coupled processes. In prokaryotes, as soon as the transcription is initiated and while it's still occurring, a ribosome can hop aboard and start translating this messenger RNA into protein sequence. Remember, the ribosome is the unusual, unique structure in the cell that's actually going to read from 5 to 3'. Remember the DNA polymerase, the RNA polymerase, they read from 3 to 5' and synthesize from 5 to 3. The ribosome, it reads the mRNA from 5' to 3'. It's unique. And how does it synthesize? It synthesizes protein. We're not going to talk about 5 and 3' because now we're referring to protein sequence. It's going to synthesize that protein from amino to carboxy terminus. So from amino to carboxy terminus is how a protein is synthesized. Now, how does the ribosome know where to bind and where to initiate translation within a prokaryotic cell? Embedded within the 5' untranslated region is something called the Shine-Dalgarno sequence. You don't have to remember the specific sequence of bases, but just remember that the Shine-Dalgarno sequence is a sequence of bases embedded within the 5' untranslated region, and this will bind to prokaryotic ribosomal RNA and initiate translation. Okay, we'll mention that fact again later, but for now, just remember the Shine-Dalgarno sequence is critically important for initiation of translation in prokaryotes. Let's highlight again once again, about polycistronic messages. This illustrates what a polycistronic message might look like. So shown above is the DNA sequence that would encode a particular messenger RNA. There's one promoter sequence, one termination sequence, and from this DNA is derived one single messenger RNA. This messenger RNA is going to go from its 5' end, which has the untranslated region, all the way down to the 3' untranslated region. You'll notice that many messages in the 3' untranslated region in prokaryotes will have this stem loop structure. There's a little stem with a loop on top of it. That simply helps transcription termination. If you're ever asked about stem loop structures and the significance of them, it's in transcription termination. So, getting back to the polycistronic messenger RNA, what you'll notice about this particular single messenger RNA species is that it encodes 3 distinct genes. Gene 1, which will have a Shine-Dalgarno sequence, then a start and a stop codon. Gene 2, which has its own Shine-Dalgarno sequence with the start and stop codon. And then gene number 3, which also has its own Shine-Dalgarno sequence with a start and stop codon. So for each of these genes, they're transcribed in one single messenger RNA unit. But notice something. They're going to be individually translated. So the ribosome will bind to the Shine-Dalgarno sequence and translate this gene into a protein. And then the ribosome will have to dissociate. And then it'll have to reassociate, bind to this Shine-Dalgarno sequence, translate this particular gene from messenger RNA into protein. If it wants to do 3, it has to dissociate. It has to reassociate, bind to this Shine-Dalgarno sequence, translate gene 3 into a protein. So the point for polycistronic messages is that they're made as one messenger RNA unit, 3 to 4 to 5 distinct genes, but they're all translated independently. That's the key. Each gene is translated independently. Each of these will be translated into an independent protein, which, remember, goes from its amino terminus all the way to its carboxy terminus. That's how we refer to proteins, from amino terminus to carboxy terminus.